I've done a bunch of CGSIs, and I'm really excited to actually talk about work that has nothing to do with any of the human genetics work that I've done or the dog genetics work. I'm going to tell you about population genetics in wild and captive populations. So I got my PhD at UCLA. I'm trained in human genetics. And then these two projects that I'll talk to you about today, we do more of this in my lab now at USC. These basically were a way for me to apply what I had learned in human genetics to non-model organisms. So hopefully this is also warming you up for Amy's talk after mine, where she will also talk about non-human species. So I'll tell you a little bit about two things that we study: captive tigers in the US, and then these Ethiopian wolves. So feel free to stop me if you have questions at any time. I'm happy to try to go more biology too if you have any questions about the organisms. So I'll start by telling you about wild populations and the types of questions that we can ask with population genetics in these wild populations. For example, right, I don't think the Ethiopian wolves are writing down their history, but we might want to know that. So how can we get those answers? And then I'll tell you about captive tigers in the US, and that's more recent work that we've done. So let's start with the Ethiopian wolf. So when I started this project, I didn't even know what an Ethiopian wolf was. This is an Ethiopian wolf. To me, it looks a lot more like a fox than a wolf. And this, the great part of this project was to get the background. There were literally 2 papers on genetics of these Ethiopian wolves and maybe 17 papers total about Ethiopian wolves. So I was like, I can sit in one day and learn all these things. That's not a good thing, but it's what happened with this work. So when I started, the first thing I wanted to know is just kind of, what do we know about Ethiopian wolves? So what I'm showing here is a phylogenetic tree of Canidae. This was made with mtDNA, so mitochondrial DNA. And down here at the base of the tree is the Ethiopian wolf. And up here are dogs and gray wolves. That's probably what we're more familiar with. So dogs and gray wolves split from each other around 10,000-ish years ago, depending on who you ask. And the Ethiopian wolf's ancestor diverged from modern day dogs and wolves. It's a wolf-like canid almost 100,000 years ago during the peak of the last glaciation. So there's a lot of phylogenetic distance between Ethiopian wolves and dogs and gray wolves. And I want you to kind of keep this thought in the back of your head as I move forward. So the question is, why are we interested in studying Ethiopian wolves to begin with? So like I told you, there's not a lot of information about them at all. It turns out that they are one of the most endangered canids in the world. I would argue that they are the most endangered canid in the world, but some folks might say now, right, that it's like Romulus and Remus, these dire wolves that we created, or it might be red wolves, which is another thing that people are quite interested in. But they're one of the most endangered canids in the world. So if you're thinking about conservation and a management perspective, right, we can use population genetics to actually understand current levels of genetic diversity. And this is important because if we want to translocate individuals to try to improve breeding, we probably want to know if they're related, right? So we don't want to have inbreeding happening in a population that's actually already pretty small. So when I started this work, we pretty much assumed that this population had been small for quite a long time. And the reason why we can assume this is because they're actually pretty much only found in this area, and they've only been found in these high altitude regions, these Afro-alpine habitats in Ethiopia. So they're nowhere else in the world. And over time, since the last glaciation, their range has gotten smaller and smaller and smaller. So their range is very restricted. Also, their diet is highly specialized. They feed almost exclusively on rodents. They have a pack structure, so basically there's around 2 to 18 individuals in a pack, and there's only one female who's mating in that pack at a time. So small population size, potentially lots of deleterious variation, but obviously we didn't know anything about that at the time. So this is something that we want to learn about, and we can actually use population genetics to do that. And what you'll notice is that there's some populations here that have X's through them, and these populations have gone extinct very, very recently. So just seeing that, can anyone hazard a guess as to why there's been recent extinctions in the Ethiopian? Climate change, that can be one. Any other factors? Humans, anthropogenic factors. So actually, there's lots of anthropogenic pressure. So the population we're looking at is this Bale Mountain population. It's separated into 3 groups. And just to show you an example of these anthropogenic forces, this is one of those populations, a Soneti population. We're going forward through time to current day, and we're looking at density of wolves per kilometer. So as humans are moving into these regions, guess what they're bringing with them? Their dogs. And these wolves have never been exposed to diseases that dogs have. So population's doing great, right? Then they get rabies and crash. It's coming back, CDV, canine distemper virus, it crashes, and we just see these kinds of waves of massive changes of population size because they're being exposed to these diseases that they were never exposed to in the past. So as a population geneticist, this is quite an interesting scenario because not only do I think, oh, this population has been small for a long time, now also I can think about deleterious variation in this context of very recent crashes and small effective population sizes. So the kind of equivalent that I would put that on as far as human genetics might be something like a small isolated population. And you know, I feel like there's very few of those small isolated populations now, right? Most places are quite cosmopolitan, but this is kind of like what I learned about as a younger grad student when I was thinking about the Finnish and these smaller populations. So I thought, okay, so maybe I can port a lot of that knowledge that I have in human genetics to learn about these wolves and actually be able to inform management about what can we do if we have the opportunity. So this population is super small. The largest population is that Bale Mountain group, has 170 individuals total. And so total, like across all the populations now, I think they're floating around 300 individuals. So it's recovering, but it's not recovering quickly, and that's probably because of the mating structure too. So this is also an excellent opportunity to think about like how assortative mating might affect traits if we can get some of that data. So what we did is we got whole genome sequence data of these Ethiopian wolves, 10 individuals. So coming from human genetics, right, me too, I scoffed at 10 individuals, but right, if the population's 170, you're actually probably looking at a good representation if you take 10 of the 170 individuals. So we sequenced them to around 45x coverage, and at the time that we were doing this work, basically what we did is we pulled all the other high coverage data that we can get for a couple of wild populations. So the Arctic wolf and the Isle Royale wolf, has anyone heard of Isle Royale wolves? One person in the back who I assumed would know about them. So basically they're these wolves on Isle Royale. They're insanely inbred. So I think what's left is basically a father and a daughter. They have not reproduced in a long time. If you look at them, they look sick. Like you can see their backs have like actual like scoliosis happening. The population, they're trying to make it recover, but it just has been unsuccessful. So it's very, very small. And then we have our classic kind of breeds of dog that we know and love. So this is what we're going to compare them to. So basically what we do is we take this Ethiopian wolf data, we align it with dog, and then we call variants against the dog genome. So they're quite distant from each other, but this is the best that we've got, and it's the only annotated canid genome that we had at the time. So then the question is, if we have all this data, what can we actually learn about these Ethiopian wolves? And the first thing that we might want to know, right, if we want to rescue them is, what's their genetic diversity look like, right? Very basic question. So what I'm plotting here on the y-axis is a summary statistic called pi. You can think of it as like a proxy for heterozygosity. What I'm doing is I'm counting up the pairwise differences between individuals. And along the x-axis are all the groups of populations that I've looked at. So what your eye is probably immediately drawn to is this. The Ethiopian wolf has literally less diversity than a breed dog, than a pug. Like everybody's seen a pug, right? Its face is smushed. If we let it go in the wild, it probably wouldn't survive. They're pretty inbred, and this Ethiopian wolf has less diversity than pugs. So that's pretty concerning. If I was going to some conservation agency, they would probably be pretty sad to see this, right? Because it's like, okay, what are we working with? Like basically clones of each other? So my thought was, okay, this is scary. Diversity is really low, but really what this is a genome-wide summary statistic. I'm not seeing anything on kind of this local level or scale. But what you can do is actually look at that. So what I'm plotting here on the y-axis is actually heterozygosity per base pair in a representative individual from each group. So up top are your Ethiopian wolves. coming from human genetics, I constantly made this mistake. Dogs have 38 chromosomes, and I was always writing my scripts for like 22. And then I was like, dang it, I have a bunch left to do. But so dogs and wolves were out at 38 chromosomes. These alternating colors are representing different chromosomes. So what you can see in the Ethiopian wolf is actually there is some diversity along the genome. In fact, there are these spikes in diversity. And what you don't see are these massive tracts where there's no heterozygous sites. So what these are runs of homozygosity. And in dogs, we can see massive ones, especially in the pug. And then if we compare them to the wild counterparts, we see this kind of pattern that sort of looks like an echocardiogram is like flatlining, right? So we see this kind of flat line, a spike, flat line, a spike. And then the Arctic wolves, they're not inbred, so we see pretty high heterozygosity along the entire genome. These Isle Royale wolves, where I reminded you they're super inbred, much like dogs, you will even see whole tracts of chromosomes that are completely homozygous in these wolves, which is pretty crazy. Like the scale of like a full chromosome being homozygous. And this term, if you're going into conservation genomics, is we now call it a sawtooth pattern of heterozygosity. So any questions so far before I keep going? Because I know this is kind of out of the sphere of what people think about, so stop me. Yeah?
Is there any like intuition as why certain chromosomes in the Ethiopian wolves would be homozygous versus heterozygous? Like, is there an advantage in genetic diversity?
So a lot of people are like pretty interested in these spikes and where they might be. So it turns out if you go to any study that's like plotting these kind of sliding windows of heterozygosity, if they adjust their filtering at all, these might disappear and appear somewhere else entirely. So we try to look into them because oftentimes, right, maybe you're expecting to see a spike in diversity at some region like MHC, right? Depending on how we filtered, that would or would not be there. So we weren't able to like quite get down to like, are there advantages for certain regions being homozygous versus maybe some balancing selection happening, but we were able to do a selection scan, and I can tell you about that. Because that one I actually trusted, and we had to like layer information to get to a point where I actually trusted the result. This is a great question. Yeah? Yeah, so I basically just randomly sampled one of the 10 individuals that I had from each group. So this is just one representative individual of that population of 10. Yeah, so I just picked one of them and plotted it. So if I went and picked another individual, I might not see this spike on chromosome 3. I might see it somewhere else.
So you have any idea how variable are these spikes? Or if you like show other samples?
Yeah, so in the Arctic wolf, quite variable. In the Ethiopian wolf, a little bit less so because they share so much more diversity with each other. And I think that's where we're actually getting some of the signal for our selection scans. Because, I guess TLDR, if you're trying to do a selection scan on something that has very little diversity, a lot of the times when you do these haplotype level summary statistics, it's just absolute noise. Because you're just looking at the same haplotype over and over, and you're actually drowning out any kind of signal that you might want to pick up. Does that make sense? Yeah.
So if this is one individual, what are the different numbers? Because they're like it's either one, like
This?
one copy or two copies, right?
Oh, this is just chromosome 1, chromosome 2, chromosome
No,
3.
but I mean like the big spike in 3, is that like the one individual that has two?
Oh, it's looking at like the allele frequency across.
Across the 10.
Yeah. So, great question. So the next thing that we can say is like, okay, we've looked at kind of genetic diversity across a genome, genetic diversity that's summarized on the whole genome scale. So we have all these different summaries of genetic diversity. We can actually use those summaries of genetic diversity to learn about the recent and ancient history of these Ethiopian wolves. So one way that we try to think about recent history and what's relevant for conservation is something that they start calling, they basically call it genomic health. All that most people are quantifying is an inbreeding coefficient, but they view it as like the health of the population. So if you have a high inbreeding coefficient, you're not looking at a genetically healthy population. So all they're doing is computing an inbreeding coefficient. So here we have the 10 individuals, so one individual per row. And then what I've done is I've taken their genomes, I found these runs of homozygosity I was talking about previously, and I've quantified them in 3 separate groups that are these 3 different colors. So this kind of cream color, these are runs of homozygosity that are smaller. We often think that these are caused by just linkage disequilibrium, which we learned a ton about, right, in the previous talk. So these small ones, most people kind of throw out, we ignore. The second group, this kind of middle group, are runs of homozygosity that we think are caused by populations with a small effective size. So this is actually quite important, and I think is becoming much more relevant now in human genetics work when we can do these kind of IBD level GWASs. So these IBD WASs, if you have populations that are small, you often see a lot more shared IBD, and you can actually use these to kind of do a GWAS, and right, you're doing fewer tests if you use the IBD segments. So we think these are indicative of just having a small effective size for a long time. You can see most of the Ethiopian wolf's runs are falling into this bin, and thankfully what they generally aren't falling into is this final bin. This says inbreeding, so these really long runs are often caused by inbreeding. It's clear that this individual is pretty inbred, but we can see if we contrast it with the Isle Royale wolves, the vast majority of their runs are actually falling in this inbreeding bin. So the good news is what we can do is we can go to management and we can say, look, genetic diversity is very low, but it looks like the population is probably fairly healthy. There are a few individuals that are inbred, but maybe overall we're okay. So that's good, that's great news. The next thing, like I said, right, these wolves aren't writing down their history for us. I'm happy to chat more about this since we're limited on time. But what we can do is look at those summary statistics like heterozygosity. There's another one called Watterson's theta, and we can use that to infer demography for like long-term ancient history. And what we find basically, and all you need to take away from this is that the Ethiopian wolf population has just been contracting through time. So it's getting smaller and smaller since the last glaciation. And right now we're estimating like a current effective size of around 110 individuals, which makes sense roughly with the population size that we're sampling from. We don't expect these numbers to match exactly because remember, effective size is different than a census size because not all individuals are reproducing, but it's within the ballpark. So we found it pretty trustworthy. So things aren't looking good for this Ethiopian wolf, but I guess at least it's not inbred, right? So I want to though like end on a kind of high note for this population. So one thing that's important that we often think about is deleterious variation, right? We're interested in complex traits, we're interested in disease. Can we quantify deleterious variation in non-model species? It's hard to do. And then let's think about adaptation because hopefully something good is happening. So one way that we can think about deleterious variation is we can use some sort of annotation method to group variants as either neutral or deleterious. For us, the kind of first partition happens at just like what's synonymous, what's non-synonymous. And then we try to add in a second layer of annotation to figure out what's like neutral and what's deleterious. So for us, deleterious means we have a non-synonymous mutation and a GURP score greater than 6 because when I was in the lab, a lot of people in Kirk's group, that was the lab I came from, were thinking about like kind of what's this GURP score threshold where we can trust the result as probably being like putatively deleterious. So that's where that threshold comes from of 6. And then neutral is synonymous, GURP score less than 2. And what you can see overall, if we just go along the genome, count up the total homozygotes per individual, the Ethiopian wolves have a lot of homozygotes, right? And sadly, it looks like they have a ton of deleterious homozygous variation. And we can try a different way of counting where we count homozygotes as 2 and hets as 1. And actually, if you are a grad student, this is a great check to do if you're merging a massive amount of data at different coverages. What you want to do is this count of alleles. And what you should see is that all of those counts of alleles for neutral should overlap. If they do not overlap, that means you've done your merge incorrectly. The count of neutral alleles should always overlap. Does not matter the difference in coverage. So here I was merging like 40x data with 15x data. If they don't overlap these distributions, something has gone wrong. So it's a good check to have. But sadly, once again, what we see is there's this huge group of variants that are deleterious in these Ethiopian wolves and homozygous. So the question is, how did they get there, right? Or like, what are they? So there's a couple things that could be happening. We have a small population. If you have a small population, right, the efficacy of selection is dampened. So maybe they're actually not that bad, they're just floating around there and selection can't push them out. could be something's adaptive, right? And these have just hitchhiked up with that adapted variant. Turns out a similar thing has actually been found in poodles. So who's seen a poodle with like a jet black coat, like the inky jet black coats? They get toe cancer at a very high rate. And you never see it, right? Because their coat is inky jet black. So it turns out that mutation that causes that toe cancer actually hitchhiked to a high frequency with the mutation that's giving them that jet black coat. So you could have something happening that's similar to that. Or if you remember back to what I said before and what my group is interested in now, when we want to learn about these wolves, we align everything to a dog. The dog is super distant from the wolf, so it could be that this influx might just be variants that I've miscalled as being derived and deleterious due to reference bias. And this is a huge issue, right? Or it used to be a much bigger issue in human genetics. This is why we want to make a pan-genome, right? So we don't do variant miscalling. So it could be due to that too. So that's something that we're interested in now is like, how do we correct for that bioinformatically? Is that possible to do? So they have deleterious variation. The last thing that I'll close with on these wolves is like, okay, deleterious variation, bad, but maybe they've adapted to high altitude. That would be good, right? Like at least one thing's going in their favor. So this brings us back to the problem that came up first, which is that the Ethiopian wolf has super low diversity. So what I found is that a lot of those haplotype level scans weren't working for us. So basically what I did was this layered scan where I looked at FST, which is an outlier statistic, basically saying how different are two populations that you're comparing. Pi, which I brought up before, heterozygosity. You can look at oppositely fixed sites, so basically what's derived in one group, ancestral in another, and then just look at fixed derived sites for stuff that might be going under selection. And basically what I did was I compared the Ethiopian wolf to these three others because, right, the Arctic wolf shouldn't be adapted to high altitude, the border collie shouldn't, and I used a bit of previous knowledge. The Tibetan Mastiff is. And what's actually super interesting about Tibetan Mastiff is that you see that adaptation at EPOS1. And if you look in Tibetan populations that are at high altitude, you also see EPOS1. We posit that that was actually introgressed from Denisova. And what's cool with the Tibetan Mastiff is we think that EPOS1 came from a Tibetan wolf. So we were like, maybe we'll see something that actually overlaps with populations that are human at high altitude. It turns out that we didn't. We found this new Krebs binding protein, which is essentially a transcriptional complex. It kicks on in hypoxic conditions and basically causes more red blood cell production. And this was the only outlier that we saw across all of those scans. So we think that that's basically how it's working. And what's cool is that we know the hypoxic like kick-on factor is caused by this HIF1 alpha, which has been found in many other scans of selection for adaptation to high altitude. So in summary, Ethiopian wolves have low diversity, been small for many years, population is just contracting, but hopefully they're not all that inbred, at least in the population we look at, they don't appear to be, and thankfully it looks like they've at least adapted to high altitude. So that's great. So yay for the Ethiopian wolves, and that's the close of kind of like what can we learn from a wild population. Let's kind of switch gears for like the last 10 minutes, and let's talk about captive tigers. So this is my AI tiger, but it turns out you can go anywhere in the world and look at a tiger, and they all look like this. You would never know what its genetic background is just by looking at the tiger. Phenotypically, they all look the same. And when we were starting this work, which was done with Ellie Armstrong, who's now at UCR, at the time we were at Stanford together, we could not convince people that America had a tiger problem. So she was trying to go around and get funding for this project, and she was like, did you know that there's actually a lot of tigers in America? And there shouldn't be. And it turns out that there's actually more tigers in captivity in the US than there are in the wild, at least at the time of this project. I think they've actually seen a pretty big boost in the wild, so we're getting closer to numbers. But at the time, there were like more captive tigers in Texas than in the wild. So more tigers in American backyards than in the wild. And of course, when we had Tiger King come out, it was no longer hard to convince people that America had a problem with all these tigers being in people's backyards. And the sad part is that, yeah, there's more tigers in American backyards, and you might wonder why. So this map here is showing you tigers. The gray area is what their range used to be, and the colors are the range now. So the range has massively shrunk over time. So in the 1940s, they went extinct in Bali. In the 70s, extinct in Central Asia, 1980s, extinct in Java, and more recently, functionally extinct in South China. There are some in captivity, but in the wild, their population, no one has seen one for a long time. So their range has dropped to less than 10% of what it used to be. So the numbers have absolutely tanked. And there are currently 6 extant populations of tigers that we're able to get data from the wild. So there's more tigers in captivity than in the wild. So our question is naturally, can you actually use these tigers that are in captivity to rescue the ones in the wild, right? Like that'd be great since there's way more of them that are in backyards than in the actual wild. But if we want to ask these questions, the first thing we might want to know is like, what's the actual genetic composition of these tigers? And then we want to say, okay, if we know this, how can we use that knowledge for actual species conservation? And this is my best PI graphic I've ever made. So this is where we got our tiger samples from. So we went around the US, or Ellie went around the US. We got data from around 150 individuals from accredited sanctuaries. So these are not from the backyard breeding programs, even though some of those individuals might be from those programs, because when the Big Cat Protection Act kicked in like, I think the 2000s, like 2022, basically they were surrendering their tigers to these facilities. So we might have a Carol Baskin tiger in there, right? Or a Joe Exotic tiger. So we got that data and then we combined it with previous information from wild single ancestry subspecies. So we had around 105 of those, which actually gave us a pretty great data set of 256 individual tigers from the extant wild populations. And then we have what we call our US generic, meaning we didn't really know about their background. No one knew about their background, tigers. So we don't know what their ancestry looks like. And so that's the first thing what we want to know, because it was actually posited that perhaps these captive populations, because they've exploded, if you think of population genetics, if we have massive growth, right, that's a lot of new mutations coming into the population. So could it be that they have all these new mutations? Or could it be a lot of these wild populations are going extinct? Do they have what was termed relic diversity of these wild populations? And we actually had no idea at all what was happening when we started this project. So the first thing that we did naturally is use something like admixture, right? And we take all of our wild tigers. So here, each column is an individual. Sorry, this is small font. Down here 0, this is 1, so we can see all of our wild subspecies have a single kind of ancestry origin as we expect. And then as we slowly transition into the generic tigers, we see that there's actually ancestry from all the wild tiger populations, including this South China population. But what's cool is we see the majority of ancestry is Amur and Bengal. Can anyone guess why there'd be a lot of like Bengal tiger ancestry in this population? Thoughts? I went to this thing as like a kid, a circus. So it turns out a lot of these captive individuals are actually also coming from circuses. So that's where a lot of the Bengal ancestry is coming from. So this kind of makes sense. We have like a lot of Bengal ancestry, a lot of Amur ancestry, and then a mix of ancestry from other groups. But the other cool thing that we can ask is like, okay, if we pick out, hopefully it advances soon. If we pick out any individual and look at its genome, so these are all the tiger chromosomes, they all look like this, but we can see that the local ancestry patterns are actually quite different. We have some individuals with a lot of Indo-Chinese, we have some individuals that maybe have a lot of Sumatran ancestry, so you can't really tell what you're picking if you just go out and get a wild or a captive tiger. With the wild, we know it's likely a single origin, but with the captives, you're not actually sure what you're gonna get. And the cool thing that we like to do in human genetics, right, is think of something in like PC space, right? And with humans, I think a lot of us have probably seen the plot of like from John, where it's like we know the people's grandparents are from this region in Europe, and then here we're plotting them in PC space to say where they're from. With tigers, it turns out we see nothing like that when we look at the PCA. If you plot geographic origin, it just kind of looks like confetti, right? And it's because they're being translocated all the time for mating, because it's actually lucrative to have certain phenotypes with these tigers, like a straight white coat. And so that's why there was this massive influx of the tiger population, is because monetarily, if you could find an individual with this white coat, you could sell that tiger for a lot of money. So looks like confetti if you plot geography, but if you just look at ancestry, it reflects ancestry pretty well in PC space. So I think that's pretty cool. And then the next thing that we could learn is just asking, like, are they this reservoir of unique genetic diversity? And we can do this with some software that's called ADS. And what you do is you subsample individuals. And then what I'm plotting here is the mean number of private alleles. So these samples are actually small when we got into like actual high quality data. What I want to draw your eye to is this. So these are the captive tigers. There's like no new alleles in there. Basically what you're seeing in the captives is coming from the wild population. In fact, like 90% of the variants that we see in the captives are shared with a wild population too. So nothing new. And if we look at heterozygosity, they're also on par with wild populations. So these are all of our individuals. We're plotting heterozygosity here. We can see there's a lot of overlap between the captives and any of their wild counterparts. So they have, which is good, they don't appear to be at least that inbred, at least in the accredited sanctuaries. So that's the next thing that we wanted to ask is actually, can we quantify inbreeding in these individuals? And the answer is yes, we can, and we're going to do it in a similar way as I did with the Ethiopian wolves. So here, though, what I'm plotting is the total amount of the genome in a run, and then the color of each dot, some of them you can't see, is an individual SNP inbreeding coefficient. So the first thing to take away is like the individuals that are the most inbred have the largest amount of their genomes in the run, so it makes sense that we're using this kind of genomic health statistic. But also the generics overlap completely with all of the wild populations. So they don't look any different from their wild counterparts. But I think the thing that's important here is, though they don't look any different, once again, here's F rho on the Y, and then down here on the X is F SNP. No matter what population of tigers you go to, if you get the most inbred individual, what you will find generally is the largest amount of putatively deleterious variation in that individual. So overall, we're seeing inbreeding depression in these populations, be it captive or coming from the wild. So to close, for the tigers at least, what we can say is like, basically if you're doing any sort of analyses on these tigers or wanting to think about conservation, you should look at the genetic composition of them. And then they cluster predominantly based on ancestry rather than geography and ancestry. And then if we look at their genomic diversity, at least in the accredited sanctuaries, similar to what we would see in the wild, and they're probably not these unique reservoirs of new diversity. And lastly, I think this is a good thing, at least in these sanctuaries, inbreeding is not as widespread as was previously thought. So I'm going to close with that and say the reason that all of this actually got kicked off is because of a tiger cub that was confiscated at the US-Mexico border. And Ollie Ryder from the San Diego Zoo wanted to know, where is this thing from? So we're actually working in partnership with US Fish and Wildlife to create a SNP panel to identify where tiger cubs are from, where confiscated goods are from, because they're actually getting everything now that the Big Cat Protection Act passed. And this tiger cub was for a rapper who shares a similar name as Tiger. So you can probably guess who that is. And we wrote this paper about building these panels. So if you're using any non-model and you want to make a panel, we have some software that can do that for you. So it was work of a lot of people, Ellie mostly, Liz and Dimitri and Katie. And then I want to say thanks to my lab for helping me along the journey. And I can take any questions if you have any left.